Today we're going to be talking about bacterial transformation. And bacterial transformation is a process by which one bacteria gives another bacteria a piece of genetic information. It's kind of like an app in an iPhone. When you download an app, a piece of information, your iPhone can now do something new that it couldn't do before. Well, it's the same way. One bacteria gives another bacteria a piece of genetic information. And that information allows the recipient bacteria to do something it couldn't do before. So maybe it can make a protein that protects it against a certain antibiotic, so antibiotic resistance. Maybe it allows the bacteria to utilize a nutritional source that it couldn't use before. Maybe it allows the bacteria to make a toxin that it couldn't have produced before. So these are some of the things that this piece of genetic information would allow that recipient bacteria to do. No matter how you look at it, it's going to give that recipient bacteria a competitive advantage. And that's very important. So let's go ahead and talk about the process here. We're going to start by drawing a bacteria. So there's our bacteria. And on this bacteria, I'm going to draw some receptors. These are receptors, and they can actually bind materials. And remember, receptors are very, very specific. Now, this is going to be an old bacterial cell. And this bacterial cell, once it gets old, it starts to fall apart. Just like, somebody that was 90 or 100 would. They start to get old, the cell wall is going to weaken, and this bacteria could actually break open. And when that happens, what's inside this bacteria can literally leak out. So let's go ahead and draw some things leaking out of this bacteria. And one of the things that's going to leak out is that bacterial DNA. So there's some bacterial DNA, and it's going to break apart, and pieces of it are literally going to start to leak out. When that happens, a nearby bacteria, maybe a younger, healthier one, can actually use some of that genetic information. So I'm going to draw another younger, healthier bacteria nearby. And it's also got receptors on its cell membrane. And a piece of this DNA could possibly bind to this receptor here. So there it is. There's a piece of DNA, one of these pieces, binding to that cell receptor. And let's draw this bacteria. Here's its DNA right here. Here's its DNA or chromosome. And when this bacteria binds that piece of DNA, it can actually bring that piece of DNA into its cell via endocytosis. So let's draw one more diagram. So here's the same bacteria. And what's going to happen now is that piece of DNA has been brought into the cell. So there it is, it's been brought into the cell. And we're actually going to incorporate that DNA into the recipient cell. So the recipient cell is actually going to break a bond between nucleotides in that DNA, and we're going to incorporate this new piece of genetic information. So there it is. That would be the new piece of genetic information. And this bacteria has now been transformed. It now has a new piece of information, and that new piece of information is going to allow that bacteria to do something that it couldn't do before. And again, that's going to give that bacteria a competitive advantage over other bacteria and especially other species of bacteria.